So welcome to Zoom a Scientist. My name is Ashley and I'm the Watershed and Lake Education Coordinator, and I'd love to welcome you to today's session. This series is sponsored by Lake Champlain Sea Grant, UVM Extension, and SUNY Plattsburgh's education program known as Watershed Alliance. Lake Champlain Sea Grant develops and shares science-based knowledge to benefit the environment and economies of the Lake Champlain Basin. As the Watershed Alliance program, As for the Watershed Alliance program, it is the Lake Champlain Sea Grant's education program that aims to reach K-12 students and their teachers throughout the basin. Our goal is to increase awareness of watershed issues in Vermont, in New York, and throughout the entire basin. The Zoom a Scientist series was created in response for the need for some virtual science programs. So rather than our traditional in-field programs, a way to engage students online. Every Tuesday and Friday from now until June, we'll be hosting a guest scientist to tell us more about their work and research in the Lake Champlain Basin. Just as a heads up, the webinar is currently being live streamed to our YouTube channel. If you're joining via YouTube, again, please feel free to use that chat box. We'll be monitoring it. So if you have any questions, go ahead and post them in there. And then after the session, the recorded presentation will be put up on YouTube as well. So if you missed something or you want to go back and see it, it'll be there. All right, so let's dive into today's session. Let me introduce our guest. So Dr. William Breck Bowden is the Robert and Guinevere Patrick Professor in the Watershed Science and Planning Department here in the Rubenstein School of Environment and Natural Resources. He teaches undergraduate courses in environmental science curriculum and graduate courses in aquatic ecology and watershed science at UVM. He is the director of the Vermont Water Resources in the Lake Study Center and leads the Vermont component of the Northeast States Research Cooperative. In addition, he is the chair of the Technical Advisory Committee for the Lake Champlain Basin Program, as well as the director of the Lake Champlain Sea Grant Program. In addition, Dr. Bowden leads the Arctic Long-Term Ecological Research Site as a part of the Long-Term Ecological Research Network of sites established by the National Science Foundation. His research site is located in the foothills of the Brooks Range on the North Slope of Alaska and is based out of the University of Alaska's Toolik Field Station. The goal of the Arctic LTER is to understand changes in the Arctic system at the catchment and landscape scales by studying the ecology of the surrounding tundra, streams, and lakes. So for today's session, Dr. Bowden is going to share a little bit on how do we gather data on water quality. So sharing their extensive expertise in water quality monitoring, including how researchers measure water quality, traditional sampling methods, and how new technologies are providing insight into aquatic systems, featuring some specific examples from a local project, the NewerNet project, focusing on land use impacts in nitrates and dissolved organic carbon loading. So without further ado, I'll turn the screen share over to Dr. Bowden. Welcome, virtual round of applause. We are so psyched to have you here.
So let me see. Are you seeing my presentation?
Not yet.
Let's try that again.
This seems to be great.
Are you seeing it now?
Yep, we can see it.
Okay, great. Ashley, thanks for that introduction. I should update my biography. One correction: I'm no longer the chair of the TAC, just a member of the TAC. For the
okay. I pulled that from the Rubenstein website. Sorry.
Want to thank everybody for joining us here today. You've probably been hearing a lot about water quality and maybe wonder how water quality is measured. A lot of people sense water quality as being a nuisance amount of algae that are in the water, in the lake, or in a river that might interfere with your swimming, the aesthetics, how the place looks. And those algae are actually natural. They're present in the water anyway. But if they get to growing very, very quickly, we can get what are called bloom conditions. And it's those bloom conditions that people sense as being a low water quality, impairing the water quality. And so what is it that causes those bloom conditions? It's the nutrients, the food basically that the algae need to grow. So when we get high levels of nutrients, we often get high levels of these algae. That's a whole nother story. What I want to focus on today is how it is that we make those measurements of nutrients that lead us to a better understanding of the water quality in the system. I'm going to pitch my presentation today to those of you who are in middle school and high school, but I think that I've got plenty here that's going to be of interest to our adult learners as well. What I want to talk about is a class of analytical techniques that we use called spectroscopy, and I'm going to explain what that means a little bit later. But this is a method that's based on the transmission of light through water. And so we're going to be examining some of the properties of light transmission, light absorbance, and how that's useful to us in determining water nutrient conditions, which then lead to a better understanding of water quality. So I want to start with a really simple question here and just pose the question, why is it that this pitcher of Kool-Aid appears to be red? Now, one simple answer is, of course, somebody put some dry Kool-Aid mix in there, which was a red color, into the clear water, and that's what made it red. That's really not kind of getting at the heart of the question of why this solution is red. Why isn't it green? Why isn't it blue? What's going on here? So for us to understand this, we need to think a moment about what color is. So our eyes see color, our brain perceives color. Color is a part of what we call the electromagnetic spectrum. There is a spectrum of wavelengths ranging from short wavelengths. So a wavelength is defined as being from the top of one cycle here to the top of the next cycle. And it's a continuum from short wavelengths to long wavelengths. These short wavelengths are things like gamma rays and X-rays, like you might get if you go to the dentist's office. And the longer wavelengths are things like microwaves and radio waves, the things that allow you to pick up a channel on a transistor radio or on your TV. In the middle here somewhere are a series of wavelengths that run from about 400 nanometers to 700 nanometers that are what our eyes perceive as being color. So these somewhat longer wavelengths are in the red region, the somewhat shorter wavelengths are in the blue region. And these wavelengths are really quite small. We're talking about thousands of times smaller than a human hair. Okay, so that's what we mean by color. And you know this, you know that the sunlight that we see, what we often call white light, is actually made up of all of these individual wavelengths of color. And you can see that in a prism. If you take a prism, it will bend the white light and create the separate colors that you see here, ranging from the red to the violet. You're also aware of these separation of colors when you see a rainbow after a rainstorm. That's the same idea of the white sunlight being separated into these component colors. And if you look at that rainbow, you'll see that it's organized from those red to the blue colors, just as we saw in the spectrum. So first useful fact for us to remember is that white sunlight is actually made up of many wavelengths that our eyes perceive as being color. Okay, so how is that useful to us? So we need to understand, first of all, what we mean by light absorption. So let's consider this clear glass of water. What's going on here? Sunlight is shining on this glass of water, and we now know that that sunlight is actually made up of a whole bunch of wavelengths, all of the wavelengths actually in the spectrum that our eyes can sense from red to violet. And as it passes through the glass of water, nothing really happens to it. And so we see white light. We see that the solution appears to be clear. Well, what was happening with the red pitcher of Kool-Aid? Again, the sunlight was shining on this pitcher, all of the wavelengths together, but there's something that's in the dye, there's something in the material that's in the Kool-Aid that is blocking, it's actually absorbing all of the wavelengths that are in the yellow, the green, and the blue, and the violet categories, so that the only thing that comes through this solution is the red color. And that's why it appears to be red to us. The key thing to note here is that all of the other wavelengths than red have been absorbed. That's why this solution appears to be red to us. So, useful fact to remember number 2, solutions like this Kool-Aid appear to have a color like red because all the other wavelengths have been absorbed, okay? So let's think about this a little bit more deeply. What would we observe if we were to add more or less Kool-Aid mixed to the recipe than was actually required? So that's getting at the notion of concentration. Concentration is the amount of something that's dissolved in a volume of water. It's literally like the dry mix, a spoonful of the dry mix that you pour into a quart of water. The technical definition of concentration is that it is a mass, an amount that's added to a volume of a liquid, in this case, water. And a typical way that we express that is grams, which are a unit of mass, per liter, which is a unit of volume. And a gram per liter would be like the weight of 2 or 3 garden peas dissolved in a quart of water. That would be kind of the sense of the mass that we're talking about. The sort of units that we typically talk about when we're talking about water quality, however, are micrograms per liter. So that's a millionth of a gram, a very small amount of material dissolved in the water. Pardon me. So let's do a kitchen experiment here, and I'm going to just use some ordinary food coloring here for this experiment. In my experiment here, I've put 1 drop of the food coloring in the glass, the second glass here. There's no drops in the first one. I've put 1 drop, 2 drops, 3 drops, and 4 drops of solution. And I've created this concentration series of blue dye. And so you can see pretty clearly that if I put higher and higher concentrations, more drops of the blue dye in the solution, the color stays the same. It's still blue, but the hue darkens. It gets to be a darker and darker blue. So useful fact number 3 to remember here is that if you dissolve more material in the water, if you increase the concentration, then more light is absorbed and the solution appears to be darker. Let's remember here, this is what's critical to spectroscopy. It's all about the water, it's all about the color or the wavelength that is being absorbed, not the one that gets through. So it's about the removal of all of these other colors which create the blue color that we see. That's what's critical about spectroscopy. Okay, so here's our first polling question, and this one should be easy. What's the best explanation? What's the best explanation for why these solutions that I've just been showing you, the solutions of dye, appear to be blue? Is it because the dye causes a chemical reaction that emits blue light? Is it because the blue is just supposed to be blue? Is it because other wavelengths except the ones that are the color blue are absorbed by the solution? Or is the blue color caused by reflection from the blue sky? Great to see these answers coming in and to see that everybody has been listening attentively.
That's great. We'll give this a couple more seconds. And just a reminder, if you do not see the poll popping up, you can go ahead and just toss your answer right in the chat box and we will take it into account there. Awesome. All right, here are our results.
So everybody got an A. That's fantastic. Okay, do I stop sharing or do you stop sharing?
That's me. You're all set.
Okay. All right, so let's go to the next slide. I'm Still seeing the poll. Do I? Oh, I see. Okay. So the correct answer, of course, everybody got it. It's the wavelengths are absorbed. So now you know that for any colored solution, you see that color because every other color is being absorbed. All right. So let's go on to our next. So let's go on to another thought experiment here. So here's another material. This is isopropyl alcohol. It's the sort of thing you might have. It's rubbing alcohol. You may have this in your medicine cabinet at home. And it's a clear solution. So here I've gone again and I've added 0 drops, 1 drop, 2 drops, 3 drops, and 4 drops of isopropyl alcohol. So the concentration is the same. The concentration series is the same, but I'm not seeing any color here. So why is that? I have material in here. It has a concentration, and that concentration is the same concentration as the blue food coloring. Why is it that I'm not seeing any color? So here's our second poll. Why do these solutions appear to be colorless despite the increasing concentration of isopropyl alcohol? Is it because a cloud passed overhead and so there's no blue sky to reflect? Is it that alcohol doesn't have any dye in it? Is it that all the wavelengths of the color are totally absorbed and so no color is favored? Or is it that pure isopropyl alcohol does not absorb any colors and so all the colors pass through it, no matter what the concentration is? What is the best answer?
So you should be seeing that poll popped up. Oh, yep, here we go. All the answers are rolling in. And if the poll is not working for some reason, go ahead and put your response in the chat box. Looks like we're split a little bit here.
Yeah, we gotta
give folks a few more seconds. All right, I'm going to share these results.
Okay, great. So the correct answer is actually D. The pure isopropyl alcohol does not absorb any colors, so all the colors pass through it. It's not answer C, because if no colors came through, it would appear to be black. We would have no light coming through. So it can't be C. The correct answer is D, that the alcohol, there's something inherent, there's some property about the alcohol that it is unable to absorb any of the wavelengths. So now we have two different substances, one a dye and the other the alcohol, one of which can absorb, just inherently has the property to absorb some colors, making it blue, and the other that can't absorb anything at all. So different materials dissolved in the water have different inherent properties or different abilities to absorb colors. So that's a really important thing for us to remember. Now, here's the third and last thing that I want to put to you as a question. And this does not have a poll associated with it. I want you to just sit and look at these two glasses a moment and ask, in which of these glasses is the concentration of the blue dye higher? Is it higher in the left, based on what we've learned so far? Is it higher on the left or is it higher on the right? Take a moment to think about that. Is the concentration of the blue dye higher on the left or on the right? Well, I'm actually tricking you here, because the concentration is actually the same in both of these glasses. You can see that when we look at them from the side. But when we look at them from the top, we're looking through a deeper depth of water. There's a greater path length that the light has to travel as it goes through the glass on the right-hand side, which is deeper than it does on the left-hand side. So even though when we look at them from the side, we can see that the concentration is actually the same, when we look at them from the top, that longer path length on the right makes it look darker. So we've learned three critical things that some scientists who were working in the early 1700s and the mid 1800s realized about light and its behavior in solutions. Augustus Beer and Johannes Lambert were philosophers, scientists who were working 250, 300 years ago, and they noted these properties. And the conclusion that they came to is the same conclusions that we've just come to with our kitchen experiment here. And that's that the amount of light that's absorbed out of a solution is a function of the inherent ability of the solution to extinguish the light. They use the little Greek symbol epsilon to represent this. And that's the difference between the isopropyl alcohol and our blue dye. The blue dye just inherently can absorb those other colors. The isopropyl alcohol can't. It's also a function of the path length that the light has to travel. And of course, it's a function of the concentration of the light extinguishing material that is in the solution. Now, here's where things get interesting to us from the point of view of spectroscopy. If we can hold these two things constant, so in other words, if we are working with a particular solution, it will have a constant value of its extinction coefficient, its ability to extinguish light. And if we always use the same size cup to contain the solution, then we can control the path length. And what that ends up meaning is that the absorbance is just proportional to the concentration. It should be just a linear relationship. And so your eye is telling you that already. If we formalize this into a graph, we can think about that solution that we saw from no drops to 5 drops and how it got bluer. And we can actually express that as a graph, a physical graph. So here we have got 0 drops, 1 drops, 2 drops, 3 drops, and 4 drops of our dye. That's acting as a surrogate for our concentration, what we mean by concentration. And then over here on the y-axis, we have the blueness of the solution from none to some to most. And we see that it's a straight line. As a matter of fact, a question that I might want to ask then is, well, is there some way for me to standardize this? Is there a way for me to actually get an exact number for the concentration? Is there a way for me to get an exact number for the absorption? And this is now where spectroscopy and the instrument that we call a spectrophotometer comes into play. Let me break this word down for you a little bit. There's 3 pieces to it. One piece is spectro, one piece is photo, and one piece is meter. So something that is a meter is something that we're measuring. It has to do with light because it says photo and light, and it's in some way breaking apart that light into a spectrum, the spectro part of this. So a spectrophotometer is a device that allows me to measure a piece of the spectrum of light. Spectrophotometers can range from being fairly simple to quite complex instruments, but when you break it down, they all have exactly the same components that are depicted in this little cartoon. So there's a light source that has white light that is beamed through something that acts like a prism. Technically, we call this a monochronometer. And the reason for that is what we're doing is separating the white light into all of its pieces, but we are then selecting a single wavelength that we are interested in, a single color that we are interested in, and we're going to pass that light through a special cup. This cup is called a cuvette, and it's special because it keeps the path length constant. The path length will always be this length. So the wavelength that we're interested in is passed through the cuvette, which has our sample solution. The light is absorbed, that wavelength is absorbed, and we will see either a greater or a lesser amount of light on a sensor, this thing that we call a photoresistor. That signal from the photoresistor is then amplified through a circuit, and we're able to read it as an output on the instrument. So we now have a way to get an exact correspondence between concentration and absorbance, and it allows me to create a thing called a standard curve. So what I do is I go into the laboratory, and using a recipe, I make up several concentrations in several solutions of increasing concentrations of the thing that I'm interested in. So let's say that it's phosphorus that I'm interested in. So I make up a solution, one of which has no phosphorus in it, and then I make up one that has 5, 10, 25, and 50 micrograms per liter of phosphorus in it. I then put each of those into the spectrophotometer, and I'm able to read the absorption that I get. That allows me to plot this graph where I'm plotting the concentration and the absorption, the concentration and the absorption for each of those solutions. For those of you who may be a little bit more advanced, I can actually use a statistical technique called linear regression, which allows me to calculate the mathematical parameters that describe this straight line. But I don't need that. I can just use the graph to get an approximation of how to use this standard curve. So here's poll number 3, and we're actually going to use this now to make a prediction. Let's say that I've now developed this standard curve. I've made my standards, I've measured their absorption, and now I've gone out and I've taken a new sample out of the lake, and I put it into the spectrophotometer, and I find out that the absorption is 0.57. If it's 0.57, what is the most likely concentration of phosphorus? And how are you going to know that? Is it 1, 15, 35, or 50 micrograms per liter? And Ashley, you may need to, you may need to move the poll aside. You can take your cursor and move the poll aside so that you can see the standard curve. It'll be easier for you to maybe see what's going on. So what's the most likely concentration? 1, 15, 35, or 50, and why? Well, you can't answer the why, but we'll show you the why in a moment.
So this is, I'll give you another couple of seconds on this one, since you might have had to slide the poll over. And again, if you're not seeing the poll pop up, if you've lost it, just put your answer in the chat box here. All right, let's see, share our results here.
So that's terrific. That's terrific. The correct answer is, in fact, the 35 micrograms per liter. And you come to that by using the graph backwards, if you will. So we know that the absorbance was 0.57. So I carry a line over to where it intersects with the line, the standard curve here, or the standard line, and then I drop down, and I can see that it's about 35 micrograms per liter. That would be the closest bet. Now, actually, if I had used the regression equation, there's a way for me to solve that regression equation backwards and solve for the concentration given the absorbance. And if I do that, I'll actually find that the exact concentration is 36.5, very close to the 35 milligrams per liter that we guessed by looking at just a visual interpretation of the graph. So that's what an analytical chemist does to determine the concentration from the absorption of the solution. Now, at this point, I do need to make a little footnote about reagents, because it turns out that many substances like phosphorus in water are actually colorless. And so they behave a lot like the isopropyl alcohol condition that we saw earlier. To make them colored, it's necessary for me to add a special chemical that's called a reagent. So reagents react with the phosphorus that's in the water to make a color. But then after that, everything that we've said before is true. The more color I see after adding this reagent, the higher the concentration of phosphorus that I see, okay? Just as a further aside, one of the difficulties of using reagents is that many of these reagents are hazardous materials, and so we generate hazardous waste in our water labs, and that's something we need to be very careful about. So how do we then put all of this together? How do we go from taking samples to having knowledge that's useful for us in management and policy? So what happens in this sequence is that someone goes out and samples the water. And usually you don't go out and take just one sample. You have to drive to your sample site or travel to your sample site in some way. And you probably have a plan for sampling many locations. And so you don't have enough time in the day to actually analyze the samples too. So you have to store them for a period of time, usually to keep them from degrading. We want to put them in a refrigerator so that they don't get degraded. Then maybe a few days to a few weeks later, we have an opportunity to analyze them. Once we get the data, that has to be summarized so that we can actually calculate from the absorption values, how do we get the actual concentration values. We then need to summarize all that data into a database, or organize it into a database. And finally, once we have it in the database, we can begin to synthesize that, and it eventually becomes a part of a report that we're able to read. This is a process that can take a long period of time. It may take weeks for us to go from the sampling to the analytical phase. It may take us months to be able to go from the analytical to the organized database phase. And it may take years for us to get from the organized database phase to a synthesis of the data. But in the end, we end up with an informative graph that tells us something about what's going on in our water body. In this case, what's going on in Lake Champlain. This is a figure that is out of the State of the Lakes, State of the Lake report, produced by the Lake Champlain Basin Program and its partners, showing the annual mean phosphorus concentrations by lake segment. So all these different circles and graphs are different locations within Lake Champlain from 1990 till 2017, almost 30 years, one of the longest records of this type of data in the country. The thing that I want you to realize is that this graph is made up of hundreds, perhaps thousands of individual water quality measurements of phosphorus that have taken thousands, maybe tens of thousands of person hours to put together to create this one graph that is really helpful to us from the point of view of the management that we might want to implement and the policies that we want to implement. All of this based on individual measurements of individual water samples. So you might wonder, is there an easier way to go about doing this? And there are some exciting new things that are happening. We refer to the development of what we're calling next generation sensor technologies. And this is something that I and many of my colleagues here at the University of Vermont, as well as others around the country and around the world, are working on. These are sensors that have a number of really interesting advantages. They can be placed out in the middle of the lake or in the middle of a river, and they can sit there 24/7. They can work all day long, 7 days a week, holidays, weekends, it doesn't matter. They sit there and make their measurements. They work autonomously. They can work by themselves. We don't have to, it completely eliminates sample handling and storage. The analysis is done right there in the middle of the lake, in the middle of the river. It can provide a data point every 15 minutes if we want to, and it doesn't generate any hazardous waste as we would in a laboratory. So this sounds great. It sounds perfect, but there are a number of problems. One is that these instruments at the moment are very expensive. This particular instrument called an S-can spectrolyzer cost a little over $20,000 for a single one of these instruments. It still needs to be calibrated in the old way. And there's only a limited number of things that we can measure with this particular instrument. As a matter of fact, phosphorus is not one of the things that it's good at. It's particularly good at measuring nitrate and dissolved organic carbon. And we still have to summarize a lot of data. But these things still have a lot of promise. These instruments have a lot of promise. So let me give you one illustration about why high frequency sampling using these sorts of instruments is useful and important. So let's imagine a kind of a standard sampling regime that you might implement if you were monitoring a lake and you were only able to go out once a month. You might get a pattern. In this case, this is a pattern for, these are real data actually, for nitrate, in this case in milligrams per liter in river water. And if we went out once a month, we might get a pattern that looks something like this. And maybe there is something going on here. Maybe that's just regular variation. It's a little bit hard to tell. But what if we were able to go out and sample weekly? We begin to see here that actually maybe there's a pattern going on here. There could be a peak. Something was causing this to go up and down. It looks like maybe there's a process that might be going on here. If we are able to collect 15-minute data, we actually see that there's tremendous information that is in this record. We're seeing all sorts of dynamics that we didn't see before in the monthly and weekly sampling. There are individual downward peaks. Sometimes there are upward peaks. And embedded in here, there are what look like daily fluctuations in the nitrate concentration. And this opens up all sorts of questions that are interesting questions for future research. So these high frequency instruments are opening up whole new understanding of the way that watersheds and rivers and lakes operate. What's the ecology and the chemistry that's going on in them that causes these patterns that we're seeing? So what is the future going to hold for these sorts of sensors? And this is really exciting. In the future, we can imagine smart sensors that are able to talk to each other. They can automatically correct mistakes that they see in the data that they're correcting. And they can send us knowledge, not just data. And this is not that far off in the future. This is actually happening in a project called the Jefferson Project. This is a project being carried out on Lake George by the Fund for Lake George in partnership with Rensselaer Polytechnic Institute and IBM, in which they are taking sensors that are deployed throughout the Lake George watershed. They are collecting information from those sensors in a high performance computing system that then produces scenarios for what is happening in the lake at that moment. This presents enormously exciting opportunities for lake management and for development of policy. And so I'm going to wrap up here by suggesting to you that there are a number of really exciting career opportunities in the future here. If you are interested in ecology, there are tremendous opportunities for biogeochemical research. That's a big term meaning the interaction between biology, geology, and chemistry in the environment, and how these things are being influenced by watershed management and the things that we do on the land. There's new opportunities for development of sensor technologies that require an understanding of engineering. There are great opportunities for data science, looking at these big data sets that we're collecting that are going to require an understanding of math and statistics. There are opportunities in complex systems science where an understanding of computer science is going to be necessary. And then we need people who are going to teach others about the science, who are going to have a background and experience in communication, and people who are going to actually help to implement things on the ground that are going to help to improve the water quality of our water bodies. And so those people who are involved in environmental management and have backgrounds in policy. So tremendous opportunities. I want to thank you for your participation. I'd be happy to answer any questions that you have. And just for fun, I want to suggest to you an advanced problem for you to consider. So I've given you some information here on the average flow of the Winooski River. It's about 142 cubic meters per second. And over a long period of time, the average concentration of phosphorus in the Winooski River is about 11 micrograms per liter. And so my question of you is, how much dissolved phosphorus does the Winooski River, just the Winooski River all by itself, add to Lake Champlain each year in pounds? Now you're going to need to use several conversion factors to solve this problem, and I've provided the ones that I think you'll need here. And I've provided the answer, but that's on the next slide, and I'm not going to show that to you. So after this seminar is over, figure out how many pounds of phosphorus the Winooski River adds to Lake Champlain, and realize that the Winooski River is only one of about a dozen different rivers that is adding phosphorus to Lake Champlain every year. And I'll stop with that.
That's great, leaving us with a little food for thought there. So I have popped into the chat box that question that Breck just posed to you in a Google Doc, so that you can click on that and spend a little time thinking and converting some of those figures later to try and figure that out. Thank you so much, Breck, for that awesome presentation, really breaking down how some of those sensors are actually calculating their data and thinking about the exciting opportunities they pose for the future. So one question we've been asking folks right out of the gate, Breck, is to just talk a little bit about how you got to where you are today, kind of what your early interest was in ecology, and then what pathway you took into your career.
Sure, I'm happy to do that. So when I was younger, when I was in middle school and high school, there was already an appreciation that nature was a beautiful place, and that national parks and lakes and rivers were valuable and fun places to go to. But already in the 1950s and the 1960s, which I'm afraid to say is the period of time I was growing up a long time ago, we were also beginning to understand that our environment was being degraded, that there were industrial activities, there were farming, agricultural activities, and there were activities just due to residential developments that were degrading the environment, and in particular degrading water quality. I used to love to play in a little stream that was in my neighborhood. When I was a little kid, I would go down and build little dams and play with the critters that I found in the stream. So I always had an affinity for water and for lakes and rivers. And so when I saw as a young person lakes and rivers being degraded, I just became interested in how and why that was happening. And I became interested in the science of it. And so that's what led me to think about the intersection between analytical techniques that we could use to quantify what's happening to the environment and better understand how we could manage it and have that feed into policy that we can use to improve water quality in the future.
Great, that's great. And then there's a question here in the chat box that's asking, when did we first start to develop these technologies and how have they developed through the years?
So an understanding of spectroscopy actually and light penetration, as I mentioned in the presentation, actually dates back to the early 1700s. And then Lambert and Beers were two scientists who thought even more deeply about this in the 1800s. And we were beginning to understand more about chemistry. Alchemy was going on in 200 or 300 years ago. But that became formalized in the 1900s. We began to develop instruments that were capable of measuring light. And so this field of spectroscopy began to blossom in the 1950s and 1960s. And it has just ramped up exponentially as we have developed better and better understanding of electronic circuits and sensor technologies and computer technologies that can interface with these sensors. There's been some very rapid developments in spectroscopy just in the last 5 or 10 years, but it's a field that actually dates back to the 1700s.
That's great. And then I have a follow-up question for that. Looking at the graph that you showed by Matt Vaughn and looking at how getting different data points over different periods of time showed different patterns, I'm curious if you could speak to maybe just a couple ecological interactions that could be missed if you were only sampling once a week versus high frequency sampling maybe every 5 to 15 minutes.
One that's quite important is that when we used to go out and take sampling, take a sample on just a periodic basis, nobody wants to go out at 3 o'clock in the morning to take a water sample when it's pouring down rain outside. People would prefer not to use their Saturday or Sunday to go out and do work to have to take a sample. And so what was happening, without really realizing it or meaning to do it, we were taking samples when it was convenient to do so. Let's go out on a nice sunny day at 12 noon on Thursday and take this water sample. And the river is fairly quiet and not a whole lot is going on. But what we find is that when we have a big rainstorm, the flow in the river gets higher and higher. We have more water that's coming downstream. And we actually find that there's an increase, there can be an increase in the concentration of things that are dissolved in the water. And so we have found that a very high proportion of the total amount of material that moves downstream in a river may occur in only a very short period of time during storm events. And during the quiet periods, what hydrologists refer to as base flow, during the quiet periods when the flow is low, there's actually relatively little that's being transmitted. And so these high frequency sensors are allowing us to be present, if you will, at times when it's difficult for us to physically be there. So we can be there at 3 o'clock in the morning. We can be there on Saturdays and Sundays. We don't have to anticipate the storm coming. We have a sensor that's out there listening to what's going on autonomously all the time.
That's great. And it sounds like also it eliminates some of that sampling bias as well.
Exactly.
Great. And so we've got a question from Jean, and Jean is asking, to what extent can we measure the trends in the annual discharge of phosphate into the lake by the Winooski River? So great question based on your problem question that you posed.
Yeah, that is the exact essence of that figure that I showed towards the end, the figure of the lake that was showing the trends. And you can find that figure online if you go to the Lake Champlain Basin program and look for the State of the Lake report. You can find that exact graph there. And you're asking the well, let me see. I think it's the $2 billion question in Vermont at the moment. And that is, how soon and how quickly and how much can we reduce that trend of increasing phosphorus, or the amount of phosphorus that we have in the lake? And that is a very, very difficult problem for a whole variety of reasons. One thing that we did not talk about is that I cannot make a perfect measurement. When I go out and take a sample and analyze it in the lab, just naturally, there's the opportunity for introducing error in small amounts of error, but those errors build up so that I get statistical variability in the data. And I need to be able to see a trend in the data that is of a bigger magnitude than just the noise that's in the data. And one of the problems that we've had when we have sampled only once a week or once a month is that variation is larger than the trend. And so I'm unable to pick the trend out of the variation of the data. So we either need a long period of time or we need more frequent data to be able to understand and kind of reduce that variation so that we can see the emerging trend. And ideally, if we implement good policies, see that trend go down.
Great. That's great. Well, Breck, thank you so much for sharing with us today and answering these questions from folks. We have a quick feedback poll that we'd love to hear from everyone on. So I'm going to go ahead and launch that right now. And so, let us know what you thought of today's session. We were so fortunate to have Breck with us with just such a, numbers of years in the field in water quality monitoring, and to share a little bit about, kind of where things have been and where things are heading in terms of water quality technology. So thank you again, Breck. Really phenomenal. We have thrown some links here into the chat box. I'll just point to those while you're all doing this feedback poll. So again, the advanced problem is here for you to click on. We have the State of the Lake report, which is in there, which has some of the data that Breck was referring to as well. And we will share with you just a quick blurb about our topic for next week as well. And so next week we are talking about, but how do we know? Sampling fish to understand what's happening with populations, with Dr. Ellen Marsden from the University of Vermont. And so for this session, we will be diving a little deeper into water, which is an alien habitat for humans, but information about fish is collected by remote sampling, so bringing fish to the surface to study. So how do scientists use those samples of fish to understand whether fish populations are healthy? So are they increasing or decreasing in abundance? How do we interpret the data from a few fish to a whole lake? And what new methods are being developed to observe fish? So that should be a pretty interesting session. If you're interested in fish and fish habitat, it'll be super interesting and up your alley. All right, looks like most folks have voted. So thank you so much for joining us today. We love being here with you on Tuesdays and Fridays to do a little bit of learning and share a little bit more about this place that we call home. We will see you next week. Thank you all so much for being here.